All right. Well, I very much appreciate the chance to talk to you all today, and greetings from Connecticut. So I'm going to talk to you about a system that we've started developing in the past 5 to 10 years that has to do with fibrosis. And we don't always think about fibrosis in a context of immunity because it shows up in a variety of other settings. Cancers, for example, exhibit a lot of fibrosis. And what this is a development of a lot of scar tissue and extracellular matrix with collagen, fibronectin, that's built by activated fibroblasts that can create tissue stiffness. And this fibrosis is, however, commonly associated with inflammation. So helminths can induce, as you know, no doubt, type 2 immune responses. And those type 2 immune responses, when they are particularly intense, the resulting inflammation can result in fibrosis as well as allergic responses. And so fibrosis is often seen as a deleterious side effect of an immune response, an inflammatory immune response. And it is associated with helminth infections. We can build fibrotic tissue around infected parasites to encyst them, for example. And this shows up in human health in a variety of settings. Fibrosis is responsible for cirrhosis of the liver. It's involved in heart disease. It's involved in cystic fibrosis and other lung diseases, scleroderma of the skin. Cancers involve a great deal of fibrosis. So there's a lot of interest in understanding susceptibility to fibrosis and what situations might alleviate and allow healing and resolution of the lesions. And much to my surprise, I've ended up studying fibrosis for the past 5 to 10 years because I was working on 3-spine stickleback and trying to study the host-parasite interactions of stickleback. And we rather unexpectedly realized that stickleback have fibrosis. And to illustrate this, I'm going to show you 2 somewhat gruesome videos. I apologize for this if it's dinner time where you are. This is a video of a healthy stickleback with no fibrosis. You see the liver, which is being moved around, is quite loose. There's no attachment between the organs and the body wall. One could lift up the intestine with no attachments to the body wall. And in contrast, this is a fibrotic individual. That tissue that you see, that connective tissue, is fibrosis. And in this particular severe case, the intestines and the liver are all glued together. They're glued to the body wall. And it's almost impossible to open the body without tearing the organs. The fibrosis is so severe. I had actually seen this for many years without recognizing what it was. I would collect stickleback in the wild. I'd say, oh, it looks like this fish didn't preserve very well. And I would blame it on not freezing or not getting formaldehyde in quickly enough. But it turns out it's actually a phenotype of interest. And we have done histological sections with trichrome stain to confirm that there's extensive fibrosis. Here's a healthy fish with the intestinal wall, that's the muscle there. Here's an unhealthy fish with the intestinal wall. And all of this purple tissue here is fibrotic lesions connecting the intestine to adjacent organs. The key thing that I want to drive home is that fibrosis is common but not universal in stickleback. The dots here on this phylogenetic tree are populations, mostly lakes, some in Alaska in blue, some in British Columbia in green. The red dots are populations with high prevalence of fibrosis, defined here as more than 30% of individuals are affected. The black dots are populations with low fibrosis, typically 0% or a small percentage. The high fibrosis, often it's 50 or 100% of fish have fibrosis. Why is this variation occurring? You can see quite clearly from this phylogenetic tree, which is based on whole genome sequence data, that there's been a lot of repeat evolution of fibrosis, but it is not universal. Why is that? Well, to answer this, I'm going to focus in on 2 populations for a lot of this talk. Gosling Lake and Roberts Lake are 15 kilometers apart. They are the same size. The fish in these lakes have very similar diets. But Gosling Lake has a very high prevalence of this tapeworm, Schistocephalus solidus. Typically, on the order of 50, 60, 70% of fish carry the tapeworm on any given year. Roberts Lake nearby, I used to say I've never seen the tapeworm, but actually this year we collected 1 individual fish with Schistocephalus. when we experimentally infect fish from these two lakes and their hybrids, we find a lot of variation in tapeworm growth rate. The high prevalence lake, Gosling, allows rapid tapeworm growth. The low prevalence lake has tiny tapeworms. The tapeworms in this picture are full siblings. They're all 42 days post-infection. The difference is the genotype of the fish that they came out of. And that's shown here on this log scale. The high prevalence fish have large tapeworms. Low prevalence or absent fish, they get the parasite at a slightly lower frequency, but the parasites are almost undetectably small. This is a quantitative trait controlled by host genotype with F1 hybrids and F2s intermediate. And I'll get to the quantitative genetics in a few minutes. The key thing that I want to drive home is when we were doing these experimental infections, we realized that we saw fibrosis. We only saw it when we exposed fish to the tapeworm, not always if the tapeworm succeeded, but fish who never saw the tapeworm don't get fibrosis in the laboratory. And it's genotype dependent. So we have a genotype by environment, tapeworm exposure interaction, where the low prevalence population, in this case, the back crosses to them, have a high prevalence of fibrosis. The F2s have a high prevalence of fibrosis, particularly when the tapeworm is present, and the back cross to the high prevalence population really don't get fibrosis at all commonly. Now, that fibrosis matters. I showed you that growth suppression a minute ago. That growth suppression is largely, though not entirely, explained by fibrosis. Fish with fibrosis have smaller tapeworms than fish without, controlling for the cross type. We have the F2s in the middle. We have the Roberts back crosses below, smaller worms. We have the Gosling back crosses that have bigger worms up above. But in each case, there's a systematic tendency for fibrosis to reduce tapeworm growth rate. We see this in the wild as well. Those previous results are all in the laboratory. We go and collect wild-caught stickleback. This is data from one lake. Individuals with more intense fibrosis have smaller tapeworms than individuals without fibrosis. So again, consistent with the laboratory results that fibrosis is associated with growth suppression. It is also associated with mortality of the tapeworm. This is a tapeworm shown here that's been encased in fibrotic tissue. That tapeworm is dead when we assayed it. We often find small cysts with either dead or dying tapeworms in them. Sometimes we find amorphous tissue that we can genotype to confirm that a tapeworm was present but is no longer alive. And we only see these cysts in fibrotic individuals. So it's clear that it is protective, but is it potentially too costly in certain situations? That is investment in this intensive defense also a liability? And the answer is yes. Reproduction is suppressed by fibrosis, not by the infection itself. So the infection only indirectly reduces reproduction in females via its effect on fibrosis. This is data from the laboratory. Fibrotic females are less gravid than non-fibrotic females. We have similar data from the wild. Again, a significant effect of fibrosis on female reproductive status. Infection did not have a statistically significant effect in the sample size. We also see the same trend across populations and in multiple populations. Also for males, males with fibrosis in the wild are less likely to be successfully nesting and defending a territory. Males with fibrosis are more likely to be free roaming, not attached to a territory. We've done experimental injections using alum that can induce fibrosis, and then we measure C-start performance in males, and we find that fibrosis significantly reduces biomechanical actions for locomotion, C-start performance for predator evasion. We also have experimentally shown that fibrosis elevates metabolic rates. So there are multiple layers of cost here that make this not a perfect outcome. So populations like Roberts Lake have defeated the tapeworm via fibrosis. They don't get large infections ever, but it comes at a massive cost to metabolic rates, predator evasion, and reproductive success. The last thing that I'm going to mention is that we have QTL mapped fibrosis and tapeworm growth. We find QTL, for example, this is a marker on linkage group 2. that's strongly associated with fibrosis. It explains about 30% of the variation in fibrosis phenotype. Inside that QTL window, shown in gray here, we then used population genomics with 100 fish per population with pool-seq data, full genome sequencing. The strongest target of natural selection inside the QTL window is right on top of this gene SPI1, also known as PU1, which has been shown to control fibroblast polarization for tissue fibrosis. So we have a QTL for fibrosis with a gene under selection that prior evidence in mice links to fibrosis, and this gene is differentially expressed between high and low fibrosis individuals in the laboratory and in the wild. We are currently doing CRISPR. We have second generation PU1 knockout fish in both Gosling and Roberts Lake genomic backgrounds, and we are going to be phenotyping their fibrosis this fall. And our hope is to confirm that PU1 actually is a master regulator of the evolution of fibrosis in Stickleback. I should also note that population genomic data from Roberts and Gosling Lake showing that target of selection has now been validated in 6 other high versus low fibrosis populations. PU1 is consistently different between high and low fibrosis populations in its allele frequency. So I'll just wrap up by saying that the intensity of immune traits can be modeled as a function of a cost-benefit function, where the benefit accrues non-linearly as you accrue resistance to a parasite, but that fitness cost continues to accrue regardless of whether you are fully immune or not. The result is a net benefit that leads to an optimum with some intermediate value, whose value will depend on the environment, the frequency of parasite exposure, other costs like predation risk, food level, other parasites. And so where we want to go next is to understand why some populations seem to have evolved a tolerance response that says, fine, infect me and grow. And other populations have evolved a highly fibrotic phenotype that is highly costly to their reproductive success. And I will end there with thanks to a large number of people who have worked on the system over the past decade. And happy to take questions.
Thank you.